Hello, everybody. YouTube tells me we're live, or StreamYard tells me we're live. So, hi, welcome back to Creation Myths. You know me. I'm Dan. I'm an evolutionary biologist. And tonight, I'm really excited for this one. I've been looking forward to this one since we scheduled it like a month and a half ago. So,
I hope I don't disappoint.
I got Mike here, Speed of Sound from Answers in Atheism. His channel is linked in the chat and also down below. So make sure that you click on that and subscribe to Answers in Atheism before you do anything else. Right now, go click on that link and subscribe because Mike does really cool stuff.
Yeah, so, as I always say, I'm going to die soon, so you got to subscribe to my channel.
Do it. You do it, folks. Come on. So we're first of all, chat. You know the deal. Just let me know if there's any audio visual issues, volume, everything sounds okay. Just let me know. Yeah. I'll be keeping an eye on it. So Mike, welcome. Tell everybody about you, and then we'll get into tonight's topic.
Well, let's see. I'm kind of, I am an amateur, but I'm a pretty serious amateur. I've been doing it for about 20 years. And I've done it all my life, basically, organic chemistry and a few things like that. About 18 years ago, I got into neuroscience and neurophilosophy and molecular biology as a sideline. And lately, molecular biology and evolution have been my main focus. But I am self-taught. And so don't expect too much. My name is Mike Bruzenak, not Mike B. You could put the whole thing there. Okay. I don't mind doxing myself because I'm too old to care about any of that. And Speed of Sound, Mike Bruzenak, Speed of Sound of Gravity, Answers in Atheism. I got all those handles. So anyway, that's me. So.
Well, I am really pumped for this because this show comes out of a chat we had earlier this summer where you kind of walked me through a bunch of stuff you've been working on with Chromosome 2. So right. I don't want to take any more time here, folks. I want to get right into it because there's some really cool stuff going on here. Basically, over the last, I don't know how many years, we think of the Human Genome Project as having finished like 25 years ago. I think it was 2001 maybe. And we've been, refining it and getting those sequences better and better since then. So we've got, the most up-to-date sequence for the human genome. And Mike has done some real deep diving into the chromosome 2 fusion site, where the two ape chromosomes that are missing from our genome, turns out you can stick them end to end together and you get our chromosome 2. So you have that fusion site right there in the middle. And Mike's been doing a ton of work figuring out exactly what is at that site, what it looks like, and what it tells us based on the latest human genome build. So I'm just going to turn it over to Mike. Tell us this story because it is super cool.
Okay, so the latest builds, the T2T or telomere to telomere builds. The thing is, when they wrote down the genome, when we had the genome from 2005, 2006. We had the idea, and I had the idea, that what you were looking at was actually what was there. Until you looked at the ends of the chromosomes and you saw a whole bunch of ends, right? Which, what's up with that? It's supposed to be ACTG. And realize that a lot has been uncovered and refined in the last 5 years, particularly, where they've done these T2T builds on both primates and humans. And the creationists have been making hay with bad information, basically, on the chromosome 2 fusion, which has been refined. And that's basically the big discovery. It's not that big a deal. But I'll show you how I did it and what it means for us. And we could share my screen, I think. Let's start doing some stuff.
There we go. Here, I'm just going to make you big. I'm going to take your label down. Give me a second here. To make you nice and big. Let's get that label out of the way. There we go. Take it away.
I always wanted somebody to make me big. Anyway, let's see. Okay, let me get prepared here. So DDX11. Now, creationists have been going on about this, and Donnie Badinsky, standing for truth, just recently did a thing called he called it chromosome 2 is agnostic. Well, it's not. because this would be a way to falsify evolution, or at least falsify our common ancestry with primates if there was not a chromosome 2 fusion. But he is right. If there is a chromosome fusion, it doesn't say that creationism is false. And that's what he's interested in. But they have gone on about this for a long time. And Georgia Purdom just did a thing on this. And can you explain this argument and why you believe it fails scientifically? So first and foremost, biblically, we know that human and chimps are not related. So, I mean, she does this. So we always start there. We always start with the word of God. And this is amazing. And this is why these guys are not science. And this is why they're not going to be getting into the schools anytime soon, hopefully, because this isn't how you do it. So she doesn't have too much to say about it, but I'm surprised that she did it and that Standing for Truth recently did a thing on it. Anyway, the most important thing to note is they've got this, all of these distractions with DDX11L2 and the degenerate fusion site and this and that and centromere and anchor D, BL30, all of these little distractions. But the reason that we believe that the chromosomes fuse is because it's synteny all the way down. And that means that if you look at the chromosomes 2A and 2B from the chimp and you line them up with the human, you get an amazing 1-for-1 crossing over between those. And I'll show you some of that. And they claim that there's, well, they have all these talking points basically that they come up with. And there's a highly functional DDX11L2 at the fusion site. And they say many, if not all, of the thousands of genes surrounding the fusion site are not found in the chimp. Many, if not all, of the thousands. There are, I think, about 1,100 that we've identified in the chimp, and they're all, and I think all but about 6 of them are found in the human. So that simply isn't true. Now, if you look at I'll show you later why they might complain about that. But anyway, so they have these talking points, and we have to go through them one at a time. And tonight we're only going to have time for one, and that is the fusion site and DDX11L2. Then my next project is to go after the centromere and that other gene. Anyway, Dan, if you have any questions or anything, just please guide me.
Yes,
I need guidance.
I will be here. I'm just going to put us back in our regular layout here. If I could just remember how. There we are. I'm going to exit solo layout temporarily.
Do you see that okay?
Yep. So I'm seeing everything fine. Chat, let us know if everything looks okay on your end, but all the sound and visuals look great to me. Yep. And I just want to point out that long list of arguments that you just showed, if you could go back to that long list, that is just, this is like, there's a human chromosome 2 specific Gish Gallop that they do whenever this comes up. Right? Yep. Where they go, oh, it's actually a and they don't even care if these arguments are consistent with each other, right? They say DDX11L2 is a functional gene, and then they go, oh, it's a long non-coding RNA. Well, which is it? Is it a protein encoding gene, or is it a long non-coding RNA? It can't be both of those things, right? They don't even care. They're just throwing stuff at the wall.
Yeah, and Donnie will say that like 5 minutes apart. He'll say that it's a highly functional helicase protein.
Yeah.
And then he'll say it's a long non-coding gene because that's Tompkins' latest spiel on it, right? And,
the key thing, I think the key thing here is something you said just a few minutes ago about this being agnostic. It's not, because creationists can accommodate a chromosome 2 fusion, right? It doesn't matter one way or the other for them, because they'll just say God made humans distinct from all the other apes, and we have different chromosomes. That's whatever, right? But it's a specific prediction of evolutionary theory, because you have 46 chromosomes versus 48 chromosomes, right? You've got an extra pair in your other apes, chimps, gorillas, right? They have an extra pair that we're missing. You look, and they all match. All the number 1, number 3, number 4, number 5, number 6, all the way to 23 and the sex chromosomes, but our human chromosome 2 is missing from their genome, and they have 2 smaller ones that are missing from ours.
Yep.
So it's a specific prediction that those 2 smaller ones, end-to-end fusion, resulted in our single larger one, right? And confirmed predictions are the coin of the realm in science, right? That is a point in your favor for your hypothesis once you have a confirmed prediction. So it's not agnostic. It's something that creationism can accommodate, but it is something that is a specific prediction that has been fulfilled for evolutionary theory. And
Right.
we all need to be clear about that.
And we found similar fusions in muntjac deer and in the babirusa pig, which are within the creationist kinds.
Yep.
So they've got to accept the fusion, that telomere to telomere fusions happen. Okay, because they think that only 2 animals popped off the ark. So they kind of got a problem there, but they never talk about that.
The deer in particular are one of my favorite examples because the 2 sister species are extraordinarily divergent in their chromosome number. One of them has, I believe it's 8 chromosomes, 4 pairs of very large chromosomes. And the other is like 40 something. It's similar to our genome. It's like, 44 or 46 or whatever it is. And they're all very small chromosomes. And clearly those are the same kind. So whether you're looking at evolution or creationism, they share a common ancestry with each other. So obviously those changes can happen.
Yeah. Yep. Now,
so take it away.
One of the big things that Donnie keeps saying, and a lot of others say, is that evolutionists are not using current research or digging into enough detail. And I'm like, right back at you guys, because we've dug in a little bit here. So I'm not sure what that is. Yeah, we got to account for a couple of things. There's a 10% loss when you put the 2 chromosomes together from, if you add up all the nucleotides for the chimps, it ends up humans have on chromosome 2 has 10% less. We have, gap differences. I'll get into that. And then the spurious exon 1 of DDX11L2, degenerate fusion site, small fusion site, all of that stuff. Erica and Ruhif both did a wonderful, and I put a link to it in the private chat. I'm hoping you put it in the description for Ruhif. He did a wonderful thing where he talks about where he shows why the site is so small and why it's degenerate. But I'll comment on that a little bit more later. But the thing without the Gish Gallup ignores totally synteny. They'll go through and they'll say, oh, there's thousands of genes that aren't accounted for. No, that's not the case. And this is a Tompkins illustration on the right here, where he drew a picture where he drew this little piece longer and he kind of like squished it in. But I went to the paper for Eunice and Prakash and I took the diagram, flipped it on its side, expanded it, and came across. And you can see that this chromosome banding, which was our first clue that we had a fusion site, right? It's identical all the way across. I mean, there's just a very slight little thing here in 2A, and we'll get into the 2A train wreck, I call it. That's an interesting little piece. But these go right across on all of them, right? And if you line those up, now there's an old tool. It's defunct, doesn't work anymore, called genuist.org. And if you go to that site, you'll find references to 2 new tools, but they told you that they're not doing the old ones. Anyway, what I did here is I took the rat and compared it to the human and then to the chimp. You can take 3 different chromosomes. This is chromosome 2, right? And you can see that the genes kind of like in the rat, they're represented in chromosome 2, but there are really a lot of crossing over here, right? And if you look from human to chimp, it goes straight across. And here's the fusion site right here, where it goes from blue to green. And that's a really important thing, is this synteny is just constant, and we can see it everywhere. Okay, it is there. And if you look at the entire chromosome, it looks like that. And you can see the rat, there's a whole bunch of blocks that cross over. And those crossovers are interesting in themselves. The fact that these blocks tend to get inverted and crossed over between whenever we separated from rats, common ancestor with rats. But there's still some synteny between the chromosomes with a rat. But with a chimp, it is just it's unmistakable. You can just see it all the way across. And let's see, this is the original picture from Eunice and Prakash that I flipped on the side. And this is the gorilla and the orangutan, chimp and man, that sort of thing. And so we have this synteny all the way down. And what I did is I took and listed all of the genes. I downloaded all of the genes and locations and RNAs and everything for the human and the chimp, chromosome 2A and 2B, from the latest T to T builds. And then I started crossing everything over. And you can see the top one here, FEM 110C, hooks up to the number 2 on the chimp. And this one hooks to number 3 and 4 and 5, and it goes all the way down. And I have those represented right here. These are all the humans, okay? This starts at the top, and it goes just perfectly all the way down until you get to, I think it's 4, okay, right here. There's a huge immunoglobulin insert for some reason in humans, that whole orange section. Some genes were inserted, but we're way at the end of the chimp 2A chromosome by the time you get to all these things that are where we have a couple of inverted blocks here, and I call that the train wreck, basically. And over here I have the chimp 2A, and this is the chimp 2B, and I have all of the genes basically accounted for. Oh, there's these areas right here, these gaps. We'll get into that. These aren't really genes that aren't accounted for in the chimp. These are locations that we've analyzed more carefully in the human than we have in the chimp. You'll find that there's big gaps in the chimp genes, and these gaps are filled in with these human, these RNs are RNAs, MT, I can't remember what that is exactly. It could be mitochondria, but there's MIRs and there's all sorts of prefixes that account for all of these gaps, because we analyze the human. We have annotated the human genome much more carefully than we have the chimp. Okay. And there's another, well, if you look at the first story, this is the original raw transcript, and you can see all of these variations where they have this FAM110C gene, and they've listed all these variations for humans. So all these things have been annotated. So you don't want to get too hung up on that. So does that make sense? Let's see here. Okay, Untitled.
It looks great to me. I'm loving
Okay.
this. I'm gonna take you down for a second. We'll just go back to this. So this is great to me. I love seeing the banding. And then what we're doing, folks, is we saw the banding, and then we're zooming in and looking at the actual sequences, right? And what you see is that you get this great match up and down that chromosome, and then you get to that fusion site, and it's all messed up where they crashed into each other, right? And then it starts lining up really good again. And so that's really cool, because it tells you something happened at that site in the middle there, and that's the fusion site. So this is great. This is super detailed, and I'm digging it.
Yep.
Chat,
and
tell us or if you have any questions.
Okay. I can show you the crash in a little more detail. I drew a picture of the crash. I call it the train wreck, but it's only like 15% of the 2A. The 2B is perfectly aligned. The 2B did not get
and that was that second one you showed where it was just perfectly green. That's the 2B, the lower chromosome, right. Chimp 13, where it just perfectly matches. So it's really all on the chimp 12, the 2A, where the disruption occurred.
Here's a train wreck. So anyway, nice. I haven't turned it into a real diagram yet, but from 1 to 467, right? But then we get down to these blocks, and these blocks crossed over, and some of them you can see inverted. the ones that narrow in the middle. There's two of them that inverted and crossed over. And it's really interesting that that happened. And you know what that is? That's proof of a fusion site, because that kind of thing happens around the telomeres. The telomeres encourage that kind of behavior. So that kind of shows you that, yeah, it's a fusion site. So rather than being a problem for evolution, it's proof of a fusion site.
Yeah, I just want to highlight this comment from Andrew here. Weird, almost like telomeric site had to degrade first before the fusion could occur. It's like,
yes,
yeah, that's the point. It's supposed to, you're not supposed to get an intact telomere at a fusion site, because the point of telomeres is to protect the chromosome from things like that.
Yeah, and I can show you an actual telomere sequence right here. Okay. And I don't know how well everybody can see it, but the parts that are kind of gray are the perfect ones, right? CCCTAA. This is an actual, this is from CHR2A in the chimp. This is an actual telomere, and you can see that they're already degraded. Telomeres are not pristine. They're better at the ends than they are, you know, toward the middle, but there's still, I think this is at the very beginning of what we've mapped with the long read technology on the chimp 2. And then if you go down to, let me see here, where did I do the, okay, this one, what I did is I replaced all the perfect ones with a P, an uppercase P. And you can see how many are not perfect. The only perfect ones are the Ps. And this is from CHR9 on the human in the latest T to T read. So isn't that interesting how degenerate it is?
It's also worth noting that once the fusion occurs and the telomere is no longer doing its job protecting the end of the chromosome, you're no longer under selection to maintain the integrity of the telomere. So you expect deletions and breakdowns of the sequence. Like, that's what we expect to happen.
And we already have breakdowns to begin with. Plus, the telomere, the two chromosomes would not have fused if the telomeres were in good shape. They were in bad shape, and that caused the fusion, basically. And then there's the complaint that it is impossible. It happened in one early hominid or primate, right? Who did he mate with? Well, I think mules have produced viable offspring, because there's a triangle during meiosis. There's a triangle of chromosomes. There's 3 of them that get together, and they repair themselves by copying the fused chromosome over onto the other side. And so that happens, and we know that happens. And we've got some other examples of where that happens. So,
folks, to be clear, this is with horses and donkeys, right? They have different numbers of chromosomes. It's off by 1 pair, like humans and chimps. And they can mate with each other, but the resulting mules are sterile. And that's usually because during meiosis, the chromosomes don't line up correctly, so they don't assort, so the gametes don't work. Meiosis fails. But what has been documented is that sometimes, instead of forming a tetrad where the 2 chromosomes are perfect and they line up, you have a sextat, I guess you would call it, where you've got the 1 chromosome, and then you have the other and the other. And that kind of forces the 2 smaller ones to line up to match up the synteny with the larger chromosome, and they can actually get stuck together. They can fuse, and then you've basically solved the problem of having the unequal numbers of chromosomes between your maternal and paternal sets, and then meiosis works properly. It's rare, but it has been documented.
Yeah. And then we have DDX11L2, which is creationist's favorite pseudogene. They love it because on early reads, they had some of the exons crossing the fusion sites, so it looked like this gene actually crossed over on the other side of the fusion site. Well, how did part of the gene come from 1 chromosome in the chimp and the other part come in the other? And they said, oh, checkmate, evolutionist. And there's a much deeper story here. But the DDX11 gene is a something you find on. Did you want to say?
Oh, sorry. No, I was just coughing.
Oh, okay.
I meant to be muted. I
DDX11,
apologize.
it's a helicase gene that you find on chromosome 12. And when you have a gene with an L and then a number, what that means is it's a paralog or it's a pseudogene. That means that they found some part of that sequence that was the same. They found some part of DDX11L2 that was the same as DDX11, the fully intact gene that creates the protein that actually works as a helicase. And the creationists insist that this is functional for some reason, but there's absolutely no information about its functionality at all until about 3 months ago. There's a paper by Zhang that came out, and it's not highly functional. They think it has some effect on sepsis, which is primarily, I think, a disease of old people. Like elderly people often die from sepsis. And if DDX11 is highly expressed, apparently there's a correlation, but it hasn't been proved and they don't know the mechanism. And it doesn't surprise me at all because every single part of your genome has some effect on some molecule somewhere in your cells. And that is how evolution works. And I want to revisit that a little bit later if we could, when we talk about gene regulatory networks and things like that. But anyway, so, but there was absolutely no evidence all these years that they're going on and calling it a highly functional gene. There was nothing. There were no papers at all. And I finally found one a few months ago. So, and it's like, whoa, there it is. Let's see here. Okay. Now let's see what's next. Okay. So here's the old 38 build, HE38. And this shows the exon on the other side of the fusion site. This blue here is the fusion site. And it pops out at 113,603,000 on the HE38 build, right? And so it keeps showing this exon on the other side, except for and this is Jeffrey Tompkins' histone modifications down here, which he says are in the fusion site, but they're actually kind of like leading outside the fusion site. There's a lot more to be said about that too, but I don't want to get too deep in the weeds on that one. But anyway, so we have this one exon over here, the first exon. This is a reverse read, right? I've reversed the DNA here. So this is exon 1, exon 2, exon 3, and exon 4 over here. And that was the first recording of the DDX11L2 gene. And how they did that is kind of a mystery. It's hard to track it down. But they had some molecular, something that they got from a lab on DDX11, and they found some similarity to this part of the gene or this part of the genome right here. And then they kind of inferred that exon 1 was there because of something called the ATGT rule, which is where you can infer exons. And they may have had a spurious read of this at some point. So they wrote it down. It was curated and then it was forgotten about. I mean, a lot of this stuff I think is automated. That these annotations, and people will just keep throwing evidence in here, and then the creationists picked up on it and took a look at it. So anyway,
yeah.
So here are the.
Before we move on from that, let me just point out that yes, going through this stuff by hand to check the annotations is extremely, time consuming and labor intensive, right? So when they're doing the sequencing, you're not really like, does that actually check out? Like when I was doing this in graduate school, my sequences were tiny. I was working with viruses where the genomes were 5,500 bases long and it was single-stranded DNA. So I didn't even have to worry about complementary strands. So I could just go through by hand, like make sure, is the sequencing valid? Is everything there? I'm doing my alignments and I can make sure that everything is what it looks like. But when you're doing the human genome, which a haploid human genome is about 3 billion base pairs, you're not going to go through that by hand. So you have shortcuts, right? You have, okay, this motif indicates this feature in the genome. wherever you find that thing, you annotate it and say, here's this. So
Yeah.
you're looking for things like start codons, stop codons, spliceosome, splice sites, that kind of thing. And that allows you to annotate where your open reading frame is, where your introns, where your exons are, that kind of stuff. And unless it's a specific gene that you're looking at, you don't necessarily have a reason to go through by hand and like check, is that actually a splice site? Is that actually a start codon? Right.
Right. But I did. So as near as I can figure, this stuff started with Costa. So Costa did this paper on DDX11L, a whole bunch of them. He found a whole bunch of them, and he mentions DDXL11. He's the reference if you look at the backstory in the genome. He is why those things ended up there. I think this Costa et al. His group here, right? And if you look, the only mention in this paper you find of DDX11L2 is right here. That's the only mention. He doesn't mention functionality. What he's showing you is that there are a whole lot of these DDX11L genes all over the genome, in the X chromosome and 17 and 12. It's filthy with them, right? So there's a lot of them. But anyway, this is where it came from. And then I looked a little bit further, and let's see what we get next. Let's see. Oh no, I took the wrong thing. The wrong thing off the screen. I'm sorry. Okay. So I was going to show you the versions. These are the different versions of the genome, and so it's really confusing when you go back and forth between UCSC, University of California, San Diego, and the NCBI genomes. They name them. They've got their own little names for them, but I primarily stuck with the T2T CHM13 for the human, which is the most current build from 2022. And then the Clint build from 2024. No, there's Pantrode. Clint was 2018. I think that's primarily what I used, because the 2024 thing has no annotation yet at all. We can't find anything on that. That'll be a thing here in a few, probably within the year, hopefully, where they'll give us even better reads for it, right? Let's see, sanity check. I don't think we need to look at that. Then we get down to, okay, I'm looking at the CHM13 T2D build for humans. Or no, I guess this is 38. Okay, I'm sorry, I'm confusing myself. Yeah, okay, here's T2T for humans, and here's the 38. So these are two different builds. This is 2023 versus 2022, I think, for the 38 build, right? Do you run into that, or have you run into have you dug into these things?
I have not dug into it in this level of detail at all. Yeah. So this is I'm learning all of this along with everybody else. This is fantastic.
Yep.
Okay. So with the most recent build, we want to zoom in on that site. And if I'm following this, we're going to look at that most recent build. And unlike the older builds where they annotated it with exon 1 on the far side of the fusion site, the most recent build
a little different.
Doesn't have that.
Yeah. Yep. Yeah, and they like if you look up DDX11, you'll say predicted to enable several functions, including ATP binding, helicase activity, and hydrolase activity. That is an exact copy from the DDX11 gene. So they didn't actually find this function at all in a lab. They copied that over from the DDX11 description, and that's why they say predicted. So they always say predicted because they don't know. They just found somebody spent 5 minutes copying that over from DDX11 to the DDX11L2. And if you go look at the other pseudogenes, you'll find the same exact copy where they're saying it's predicted to. But when you look into the gene, there's no way that it can do those things. Okay. So AGR often carries over molecular function, biological process, and cellular component Go terms from the closest protein coding paralog. That's how that happened. So you don't want to just read that and believe it, okay? Like our creationist friends like to do, because it gives them ammo, right? So they go ahead and blah. Okay, so we look at the two entries for. There are two entries in NCBI. 24004.2, 24005.3. I think 5.3 replaced 4.2, which replaced 4.1. So when they get better and better information on these things, they always go to the newer version. So you want to be careful looking at old versions. And then if you go look at this in the T2T build, we find some surprising things. Okay. We've got this one thing here where we have 2400402, which shows the fusion site. Okay, my fusion site here starts on that red line. It turns from TTAGGG to the other, opposite end, right at this purple line. And then the red line is the end of the fusion site. So this is the fusion. And here's Tompkins' little humps down here. These are histone modifications. Okay, and then we have the old gene. Let's see, we've got an old read here. No, that's wash. And this is the latest, newer read. And then we have the oldest read, 24004.2. Well, then we have something called a cat liftoff. And a cat liftoff is where they've taken the best representation that they have of the gene, and they copy it over to they try to match it with the newest genome. And it ends up at a very different position. Like, you remember the fusion site in the 38 build was 114 or 113, and here it's 114,028. So it's in a different location. So things have shifted somewhat as we've gotten a better and better read. Does that make sense so far?
It's all good to me. Chat, let us know if you're following along.
Yep. Yeah, this would be boring for some people. But then if you go ahead and look at this, there's let's see, we got the fusion site right here. Okay.
Well, hang on. Mike, can you go back to that previous slide though? Because I think you kind of buried the lead there in the actual graphic. You could see the one more forward.
Oh,
there you go.
That.
Right?
Well, I buried it for a purpose.
So, okay, cool. Just making sure. Okay, good.
We'll get there. We'll get to it. Yeah.
We'll get to it.
Yeah, like I say, I explained this to Austin, to a friend, and he said, where's your discovery? Well, I kind of went through it real fast. Okay. If you look at these annotations, right? This one right here is, let's see. Okay. It shows, so this one right here shows, I can't make that bigger. So it shows two different reads, basically, and it is showing the one exon on the other side of the fusion site, right? And if you look over here, well, there's the NR024005.3, and it looks like we still have an exon on the other side of the fusion site, right? But if you expand it, if you click on it, what it does is it separates those two. And then you can see the newest read right here. So it'll separate it. And you can see that let me see here, the 03 does not have does not have one on the other side of the, let's see, 2 does, and 3 does not have one on the other side of the fusion site, basically. And it's kind of hard to see on that, but if you go down to, let's see here, well, we'll go back up. And the important thing is, you're right, I kind of buried the lead on that. Let's see, where is it? Okay. That the cat liftoff shows the actual gene right here with the exon 1, 2, 3, and 4. And if you look at where these, basically, if you look at how it relates to the DDX11, you can look at the way the protein maps out. and you can go through in utter detail. And let's see, we go right there. We go down here. Okay, this is the actual DDX11 protein. And this part right here on the bottom is the DDX11L gene, basically. So you can see the protein is this long, this whole thing, okay, is the protein, DDX11. And then right down here, it gets cut off. And the only part that DDX11L2 is the very end, or the C terminus end of the protein. Okay, so we're looking for MSEG-INF or MSEF-INS. There's two different reads, basically, and we can go down and take a look at that cat liftoff gene, which is what I did here. And you don't find it in exon 1 at all, but you find it in. Am I still tracking?
You're good. You're
okay,
good.
There's only 127 amino acids in the DDX11L2. There are 906 in the full DDX11 gene, which is functional, right? Okay.
Right. So this pseudo gene has experienced degradation, deletions. So when you line those exons up, if you were to remove the introns and translate that gene, you would get a protein that is what, like 1/8, 1/6 the length of the actual functional protein.
Yeah, this green part down here where it starts with MSEG-INF, and this is the full helicase, and they get their name from DEADbox, which is dead, DEAD, which is aspartic acid, glutamic acid, alanine, and aspartic acid or histamine. And so you get either a DEAH, and the DDX11 gene has a DEAH, and that little guy is where the gene gets its name. And there are all sorts of things here that I've highlighted in color that are part of the molecular machinery that gives it the helicase activity, right? So we just have a little fragment down here.
And we can also note from this that the parts that are responsible for the helicase activity are absent from the 11L2 sequence.
Yep, and that's the important part. Now then, if you go ahead and look at this cat liftoff gene, what you find here is we got an I, we got a G, we got a G. Oh, we got a start codon. And then we have MSES-INF, which is that pseudogene. And that's why they place this new annotation, this pink one, that shows no exon on the other side of the fusion site. That's why they placed it here. This is the first intron, right? And the other reads are very confused when you try to find the actual similarity to the DDX11 gene, okay? And here we got it again, showing the DDX. Okay, here's the fusion site. Here's the center of the fusion site where they switch over from CCC to TTA-GGG. And here's one end of the fusion site. Here's the old exon. And you can see it here that is in the NR 24004.2 read, which replaced the 0.1. But now we have the newer one, which they say right in the documentation, this one should replace 0.2. That 0.3 replaces 0.2. It happened in like 2017. Nobody noticed.
So let's just be clear about that. Let's just be clear about this then. Any creationist that's talking about this and using the annotation that involves an exon on the far side of the fusion site, according to the people annotating the genome, that is outdated and is no longer what kind of the I don't know what you would call it, right? The master list, right? The reference genome might be a better word, right? That exon that causes the sequence to span the fusion site is no longer part of that annotation. And they say specifically in there, don't use the old version. Here's the new version. Use this.
Yeah. And I think they actually made this one up. They got a long read in some lab, maybe. And then they went ahead and used the AT-GT rule or GT-AT rule for where you find introns. And they kind of like figured out where that thing might map. And they tried to map this to to the DDX11 gene, and this is what they came up with. And they probably didn't spend more than a day doing it, right? Because they didn't care, because there was no evidence that this had any functionality at all for anybody. But then now in the latest build, if you take and look for the similarity to that DDX11 actual protein, you'll find it starting right here with a start codon, that MSS-INF, which shows you the very, very tail end of that protein. And that's why it's called a DDX11L2, basically.
Right.
You'll find those. There's a whole
again,
bunch of.
Again, for anyone who wasn't here for the beginning of this, because my goodness, a bunch of people have gotten here since then, the L means like. So it's the DDX11 like. And in this case, what that means specifically is that it's way shorter than the real protein.
Yeah. Let's see, the transition from 0.2 to 0.3 indicates that the RefSync gene model for DDX11L2 has been updated. Such updates can involve changes in the gene structure, such as addition or removal of exons, changes in splice sites, or adjustments to the gene's boundaries. These updates are part of an ongoing effort to improve the accuracy. So there it is. That's what you have to understand. What you see in the genome is not all the time what is really in our cells, right? We humans have the habit of believing anything we read. And you got to dig a little bit further into it. Now down here, I show all the different, these are all the different histone modifications here. Here's the gene up here. And here's something called the WASH2P gene, which we'll talk about a little bit. That was another discovery that I made. If you go ahead and, I'm going to tell you about it now. If you go ahead and blast this entire sequence right from here with the fusion site, and I'll show you that in more detail, all the way over here to the end of this chromosome, you'll find these things right next to the telomere in about 6 different genes or chromosomes, 6 other chromosomes. And I even found it in chromosome 2, and you'll find it on the chimp. You'll find it all over, and you'll find a whole bunch of DDX11 genes sitting there. Some of them documented, some of them not documented yet. And you'll also find this WASH2P gene, which is in our chromosome 2 right next to the DDX11 gene. So, like, these DDX11 genes, for some reason, end up in these right next to these telomeres at the ends of our genes, except for chromosome 2, where we do have one by the telomere, but we have another one right smack in the middle where we claim to have a remnant of a telomere. Why is that? Why is this block copied right here in the middle of the gene? Everywhere else it's on the end of the genes. So that's more proof of a fusion site, basically, that we have.
And this connects the specific point about the DDX11L2 pseudogene, which again, this whole family of genes, the DDX11 family, they're all found in subtelomeric regions. They're found adjacent to telomeres.
Yep.
With the exception of this one that's in the middle of human chromosome 2.
Yeah.
And it's adjacent to the telomere-telomere fusion site. But connecting this to the broader, here, I'm going to take the screen down so you can see my face while I talk. There we go. Connecting this to the broader idea of synteny that you brought up earlier, it's not just that you have head-to-head telomeres at the fusion site with immediately adjacent to that something that we only find elsewhere in the genome at the telomeres. It's that adjacent to that is something that we also find in that correct place in other subtelomere regions. So as you move away from the fusion site, it's not just this one thing that's right there, it's other stuff that you find as you go down the chromosome is where you would predict it to be if this was in fact a fusion site. So it's not just that we have a confirmed prediction of, oh, we found 2 chromosomes that line up, or we found one big chromosome that lines up with the 2 small chromosomes. It's also, oh, with the middle, you've got a head-to-head telomere, and you've got a pseudogene that family is only in subtelomeric regions. And then you've got, oh, adjacent to that, you have this other gene that's always found with it, right? You have all of these things. This is, folks, what I'm trying to get across here. This is not one confirmed prediction. This is a
Right.
whole series of confirmed predictions that all point to this being a telomere-telomere fusion event. It's all different pieces of evidence that are independent of each other, all telling you the same thing.
Right. And we're deep down the rabbit hole, but we don't want to forget the first thing. Synteny, synteny, synteny. That was the big thing, right?
Yes.
And this Costa paper, he talks about this whole family of DDX pseudogenes. And he lists a whole bunch of them, and they're all out there by the telomeres, except for this one, which is right, smack next to that fusion site. So anyway, that is important. Okay. And I did it again. I don't know why I keep doing that.
There you go.
I keep doing that. Oh, let's see here. Okay, we're good. Okay, so what do we got next? Oh, this is interesting. Okay, so this is the old up here, this blue. This is the old exon. This is the fusion site right here. You can see the TTAGGGs here. This is the whole fusion site. Here's Tompkins' first exon from the 0.2 build, which we've replaced with the newer one. And down here, I have my cat liftoff gene, which shows the read start of the first exon, the second exon, third exon, and fourth exon. And these are in pink are the exons, in black are the introns, which get clipped out, and it goes all the way through. Okay. And it shows it in utter detail. Now let's see here, CHIMP12. Okay. Now the other interesting thing is I did a blast of this whole sequence because I thought, okay, maybe there is something out there in the genome somewhere that has that first exon.
Here, hang on a second. Hang on. Before we go any further, let's just explain to everybody what BLAST is.
Oh, okay.
Just make sure everyone's on the same page with that. So what is BLAST?
I don't know what the acronym stands for.
Something basic, something
Searching
longitudinal alignment sequence tool or something. I can look it up. I forget what it stands for.
Yeah.
Search tool.
Yeah, it does a fuzzy search, basically, like a regular expression search for any of you programmers out there. And it does sort of a fuzzy job of it, and it tries to match as good as it can, and it gives you a score about how good it matches.
I got it. Basic local alignment search tool. I was off by one.
Okay. Yeah. Yep. And so I thought I'd do the whole thing and figure out whether or not this whole thing came from somewhere else in the genome, which it obviously did, because it kind of overwrote a little piece in the middle. And what I found is that starting right here, I found it in chimp chromosome 12 by the telomere, all the way from here down to there. What I didn't find is the fusion or that first exon. When I did just the first exon, I only found a little tiny 3, 4, or maybe 6 nucleotide reads. That first exon never came from anywhere else. It was created entirely with, I hate the word, but random mutation, basically. That's how it ended up there. And the reason they called it an exon is because of this. Oh, somewhere there's the, what is it? The GT. I keep forgetting that one. What is it called? The rule for exons and introns, where the introns get
the splice site. There was the splice site motif
splice
was
site,
there.
Right.
Yeah.
Yeah. It is. Let's see here. It is called GTCD. There's GTAG. So right here is a perfect one. You can see the GT right here, and then down here you can see the AG. And that's the beginning and the end of the intron. There's also another conserved part of the intron that's back here a little bit. That's kind of the pivot point. And the rest of the intron is kind of like what we would call junk drawer DNA, basically. It doesn't really matter. It's not highly conserved. But this shows you in detail what the new read looks like. Okay. And let's see here. What we got here? Oh, these are all the places where I find DDX11 lookalikes. Basically, they're all by the telomeres down here. And up here, this was a blast. and there are some little odd things in the middle. And this is the actual DDX11 gene right here. And you can basically, this is a ensemble blast, which allows you to really dig into these things and see how much of a match you have, how good it is. And here we have the chromosome 2, right smack there in the middle. There are some other reads here and there. None of them are by what looks like a fusion site. There are other DDX11 or other DDX genes mostly. So anyway, so I did that. And this again is another picture of 2 and 3. And here's 3 with 3 exons. But the new one has, the new cat liftoff has 4 exons all on the one side of the fusion site. And that's it. Okay. What else we got? Oh, here's the thing on gaps. Basically, when you look at the synteny, okay, if you analyze this, this is, let's see, thousands of genes missing talking points. Basically, we've got, all these genes on the human that is not shown on the chimp. But then if you go and look at it, there's, this amounts to 1.8 million and 1.1 million for the human. And it's like, oops, but I'm going to look in great detail at that. I'm going to tell you exactly why that is. One of the things you find here is an antisense copy of the gene. And I'm not sure if it's in a different spot and then you have some RNAs. And then you have, some other little remnants. Some of them are really tiny. Look at the size of those reads. Like 136 nucleotides for this RNA. And these are things that we've identified in the human that we haven't yet identified in the chimp. And you don't want to get excited that there's thousands of genes missing, because there aren't. Because these match up perfectly. The GCFC2, whatever. So, is that clear?
Yes, and also those are definitely not like genes because they're teeny. Everybody should understand that those are
RNA
transcripts
SNPs
that are like you'd have to look at the levels of transcription to have any confidence that they're doing anything. But at that length, I mean, you're getting teeny little peptides for those. That's not going to be any kind of enzyme or anything. That's if it's anything, it's going to do it as a small RNA. But you would need a lot of small RNAs to accomplish anything. So you're going to find a lot of transcripts that are going to be spurious transcripts because DNA polymerases are not precise instruments when it comes to picking where to start.
And that brings us to sequence logos. So these things here are the sequence logos for telomeres, basically. And what they show you is the real high ones are real certain. Like those would be conserved nucleotides. And the lower ones are kind of like, the lower it gets, the more it's like a suggestion. And it's got a lot of fuzz room, right? As to which one, this shows you the probability in 2 bits of whether you will find a cytosine there or a thymine there, or maybe a cytosine here and maybe a thymine. So it shows you that these are really short, basically. If you look at, maybe I'll defer that, but I've done a real deep dive into gene expression. And gene expression is just prolific. I mean, it's not the word I'm looking for. I'm looking for a word that, and I don't want to say it's oh my God, I can't think of the name. Anyway, it's really easy to happen. It's really easy for a transcript to get started, for the machinery to lock onto the gene, for the histones to be modified in exactly the right way. So you'll end up with little snippets here and there. And our cells are filthy with junk to our DNA. And that's how evolution works.
And biochemistry is super messy. And I love figures like this, like you can see on screen right now, because it shows you, it's such a great illustration of how silly things like, oh, that sequence is super improbable, like the Douglas Axe 2004 paper, or any like irreducible complexity thing that are based on it has to be this specific sequence. Because you look at stuff like this, and even for very important, very tightly controlled sequences, there's always variation in there that you can have where it does the same job approximately as well across,
Yeah.
various sequences. So it just seeing figures like that where it has the heights of the letters, almost like a bar graph where each letter is the height scaled to like its frequency at a particular site. It's super cool to see that because you see just how much variation there is. And if you look, for example, at polymorphisms within the human genome, or you compare the human genome to the chimp genome and the gorilla genome, you can generate figures like that. And obviously, if you take a certain gene, the human version, the chimp version, and the gorilla version, for example, there's going to be a little bit of variation in there. They all work fine. They all do their jobs in basically the same way, right?
Yeah.
Human physiology and chimp physiology and human biochemistry and chimp biochemistry, pretty much the same. So, having those little variants in the sequences is a very clear illustration that undermines this kind of whole set of creationist arguments about the necessary specificity for this kind of stuff.
Yeah, and machine-like behavior,
Yeah,
not machine-like at all.
not
This
at
is
all. It's super messy.
the Tata box, basically. It's about 7 long, and this shows you TA, and then there's like an AT, and then that has to be an A, but that could be a T or an A or whatever. And then these are the other ones. There are other things that start the process, the BRE and the INR, and they show you these sequences are extremely short. And if you look at this, no, that's not it. That's not it. Oh, where is it? Where is it? Okay, I think this is it. There it is. Okay, the probability of having a sequence, if you were randomly to throw DNA together, those 7 particular, like a sequence of 7 nucleotides, there's 195,000 of them that you would find randomly in 3.2 billion nucleotides, right? If you have 6, you'll find 781,000, and it goes all the way down here to about 15 long before you'll only find 3 of them randomly by random chance, or whatever the hell they like to call it. And then the longer it gets, the more unlikely it gets, right? Down here, we have a 0.0029% probability. So this is the possible K-mers. So if you were to randomly throw DNA together, helter-skelter, using dice or whatever, you would find 195,000 of those Tata boxes in the genome. So that's really important to know. And that's why basically transcription is so ubiquitous. And that's why the ENCODE project found all those hits, because it just happens randomly, naturally, that sort of thing.
And I'll just add to this. I'll just add to this because a common thing you hear creationists say with this is that energetically, that's not feasible. They wouldn't allow it, right? The cells wouldn't allow it to be transcribed if it wasn't actually accomplishing something important for those cells. But there was I think it was a 2015 paper, and Michael Lynch was, I believe, the lead author on it, where they showed that the energetic cost of transcription is below the selection threshold, whereas the energetic cost of translation is generally above it. So what that means is you're going to get a lot of spurious transcription, where you get RNA that's not doing anything, and it's typically short, typically in low copy number, typically degrades relatively quickly. But you have much less commonly junk proteins. You do occasionally get a junk polypeptide, right, where translation happens just spuriously because you get a transcript and it goes through the modification and export process and a ribosome grabs onto it. That does happen, but it's much less common because translation is much more energetically intense than transcription. So it is more likely to be in excess of the threshold for selection, meaning it causes a detectable problem in terms of fitness, and it gets selected against if it's not actually accomplishing some beneficial function for the organism, right?
Yeah.
So this has actually been shown empirically that spurious transcription, very common. Spurious translation, quite rare. And
Yeah.
the length of these regulatory sequences that's going to facilitate or prohibit transcription, like as you're saying, is mutations are just happening in regions that are unconstrained. For sequences that are that short, you're relatively likely to just stumble upon a sequence like that, or one that's differs by maybe one nucleotide. And
Yeah.
that's going to associate with a polymerase every so often. Those sequences could be longer, but you can't select for that because
Yep.
the energetic cost that you save by having a longer, more tightly controlled initiation process, and one of the steps in that would have a longer sequence that has to be specific. There's no benefit to that selectively because the amount of energy you waste by having a sequence this length that it pops up sometimes randomly, the amount of energy you save versus you waste is not a selectable difference. So that's
Yeah.
why it's this way. Like evolution explains why this sequence is the way it is.
Yep. And I've become convinced. Okay, when you look into gene regulatory networks, there's the way that we depict them in our biochemistry book. The part of the network that we've explored and that we understand, and then there's all this other junk drawer sort of stuff going on that's locking into those networks, even proteins, but mostly RNAs and microRNAs and whatever. And they all have an effect. And every now and then they give us the right kind of variation. Like that thing about sepsis and DDX11L2 that just came out in 2025. That shows you that sometimes it locks in and it does something to an immune system protein, basically, that helps us. It has some effect. And a person will survive sepsis that has basically high expression of DDX11L2, and a person that doesn't have it is more likely to be a fatality. So these things are all over, and I call it junk drawer DNA. And there's just tons of little molecules floating around, slamming in each other, and every now and then they find a pathway. And but the thing with sepsis is it's not going to get selected for because it's primarily a disease you get as you age. It's not likely to be highly selectable, right? You need something that's going to affect reproduction. Anyway,
No, that's exactly right. And I love teaching this point in class, talking about traits that manifest after typical reproductive age. You can't select for or against them because they don't affect your fitness, right? They don't affect the representation of your alleles in the next generation. And susceptibility to sepsis is one of those traits because it typically occurs much later in life. So you're not going to select for or against traits associated with that because by the time it becomes an issue at a frequency that would be relevant at the population level, everyone's going to have reproduced by that age anyway, right? And so there's no effect one way or the other. Yeah.
That's why us old people are so screwed.
So I saw a minute ago you pulled up a slide about chromosome 9.
Oh yes. Oh, we want to go there
about that. Yeah. So let me just get an idea for everybody. So we talked about synteny of the chromosome 2 with the two chimp chromosomes 12 and 13. We zoomed into the fusion site. We looked specifically at the DDX11L2 transcript there, the regions that correspond to that pseudogene in the immediate vicinity. We looked at some of the other stuff that's around that site, and we looked at kind of the history of the annotation of that sequence. So Mike just laid out for us, where are we going to go from here?
Well, let's see. Okay, yeah, we could go there. So Donnie went out and he did a blast on, I think they used blast, and I can't remember what site they used. I think it was UCSC. And they found a fusion site on chromosome 9. And it was something that, well, let's see, I've got some pictures here. Let's see here if I can find them. Well,
I'm going to put you back up big. So we're going to do the chromosome here, just as we're going here. So we're changing gears a little bit because the argument here now with chromosome 9, to be clear, is that this chromosome 2 purported fusion site is not a real fusion site because we actually find these throughout the genome. So calling this a fusion site is improperly classifying some other thing in the middle of that chromosome And Donnie Badinsky, standing for truth, for anyone unaware, YouTube creationist, he has claimed to have identified a fusion site in chromosome 9, saying, well, look, obviously this one's not real, so how can you say that chromosome 2 is real?
Let's see. Yeah, I'm trying to. I wish I had marked that one. I wasn't sure I was going to talk
here?
about
He was
that
in the
one.
chat a minute ago. Donnie, tell me if that was a fair representation.
Yeah.
If anyone else can tell me. But I think that's the point, is that pointing to, well, there's not one in chromosome 9 because, look, all the apes have the same number of chromosome 9. So obviously there's not, right, there's not a disconnect there. So if you can find all the hallmarks of it in chromosome 9, then that reduces confidence in chromosome 2. So tell us about this one.
Yep. Yeah. Okay. So if you go ahead and use this sequence right here, which is a perfect, pristine telomere to telomere sequence, right? With, I think I used 6. I used the same one that Donnie used, same exact one. Now, Erica did not. She used a more convoluted one, a more degenerate one from the fusion site. And she didn't end up with this, but you will find something on chromosome 9 that basically spans 50 characters. And it will have, and I've got some pictures of it. I wasn't quite prepared to do this one, but let's see. We'll do it anyway.
If you don't want to do this one, do whatever you were planning.
No, we're doing it. Okay, chromosome 9 fusion site right there. Okay, so Donnie found a pristine fusion site right here, right? But there are no DDX11 genes there to support it. There's, it is like really degenerate, except for this one little area where it is absolutely perfect. And that is enough characters that could not have happened randomly, right? So now either this is an ancient fusion site, or there's another phenomenon that we know about that involves telomeres and telomere repair that sticks these things occasionally into the genome. And I don't know much about it, but I did run into it, and there is some information that it can happen. So it happened in one of two ways. So either this is an ancient fusion site, or it is a accidental little chunk got blobbed in the middle. But if you look around it, there's nothing resembling a fusion site. Now, Erica and Donnie have been arguing about, because we insist that for a fusion site, we've got 892 bases. And we got a lot of these repeats basically at chromosome 2, but we only have these few perfect ones. We have a 5-2, I think it is, 5 TTAGs or a 5-3. I think there's 3 CCTCs. And then if you look at our chromosome 2 site, we've got like 1443. So it's a lot different. So it's a lot more dense, right?
So the weird thing with the chromosome 9 claimed fusion site here, and I did see Donnie in the chat confirm that I had it right. Thank you. We know that, so the chromosome 2 site, as we've been discussing, is degenerated, right? It's missing stuff. There are mutations that have occurred, and we can document that because basically once it's no longer under selection to function as telomeres, you're going to have relaxed selection on those sites, and mutations and deletions are going to occur, right? And since humans and chimps diverged 6 to 7 million years ago, give or take, we have a ceiling on the duration of how long that chromosome 2 fusion site has been under relaxed selection.
Right.
Now you look at the chromosome 9 site, and you have this very short sequence. Where'd the rest of it go? Well, all the other apes have that same chromosome 9. There's not a disconnect in the numbers like there is with chromosome 2 versus chimp chromosome 12 and 13. There's not a missing chromosomes where we could say, oh, it's these two chromosomes crashed into each other and resulted in human chromosome 9. So you have to go into the more distant past, right?
The gorilla has it too on chromosome 9.
So,
but the chimp doesn't. Gotcha. Evolutionist.
So
something weird, happened to the chimp, right?
It has to be further back. But why is it? It's a weird paradox where it's both shorter, indicating more deletions, but the sequence that's there is pristine, as though it has been preserved through purifying selection.
Right.
And you can
And
calculate the likelihood of that based on the length of that sequence. Given the length, and if it is in fact a fusion site under relaxed selection, the likelihood of that pristine site staying that way while the rest of it has gotten perfectly deleted with a scalpel strains credulity, let's say.
Yeah. And what I would do to research this is take a look at the area around it and figure out what is going on with the chimp and whether or not there's a block that got transferred over in the chimp, did not in the gorilla. I think it's evidence possibly of an ancient fusion site that goes way back to our, before our common ancestor with gorillas. I'd want to look for it in the mouse, and I'd want to, the rat, and there's a lot of research to be done there. But anyway, the reason Donnie got like hundreds of them, right? Because they did a relaxed search. And what they found is they found they found interstitial telomeric sequences, ITSs, and they found them all over because they happen all over. And there's a reason that they happen. There's a reason why we have these TTAGGGs. But what we never have, we only have in two places, in chromosome 2 and chromosome 9, is the head-to-head, right? So this is the only one where we have TTAGG followed by a CCCTC. Well, where's the good one? CCCTAA, right? So that's really important. So this is a pretty rare find. Donnie said he found hundreds of them or dozens of them. No, you didn't, Donnie. You found ITSs all over with just either TAGGG or CCTC, but never the two together. So this is the only place we found it. And this is a mystery, and it's another little piece of evolution that we have to take a look at, right? Oh, what I have here. I've got some more on. I got a whole bunch on this. Let's see, the CHR9 fusion.
Let's just summarize for everyone kind of what we've covered here, just so everyone's on the same page with us. So we covered a whole bunch of stuff with chromosome 2. Let me see my face. There we go. We covered a whole bunch of stuff with chromosome 2. Went through big picture, zoom in, history of it. Now we got chromosome 9. You have this very short but very highly conserved, potentially, sequence, but none of the other stuff around it that would indicate the presence of head-to-head telomere fusion.
Right.
Which is odd if it is in fact a fusion site, right? Now, it could be a fusion site that predates the divergence of humans and chimps and gorillas, for example. Right? I'm not sure how far back you have to get for chromosome 9 to look different, but you have to go significantly further back. Yeah.
Yep. And Donnie claims that the fact that we find them elsewhere means that the fusion site was just an accident. No.
When you say accident, you mean it was annotated as a fusion site incorrectly.
Something like that. I'm not sure where he's going with that exactly, but anyway, let's see.
It wasn't an accident in that no one said fused together. It's just like that's just a thing that happens in chromosomes.
Yep. Yeah, and then I did a little bit of looking at some of the genes around it, and what else did I do? So this is something I'm still working on. Here's our fusion site right here. This shows, what our fusion site looks like. I believe, this is ours, or is this Donnie's? No, this is ours. Okay, this is a pretty good. No, pretty good.
Mike, your face is on screen, not your screen.
Oh, okay. Wow. We don't want to look at that. Oh, there it is. Okay. So this is our fusion site.
There we go.
Or no, this is Donnie's 5.2. Okay. Mm-hmm. This is Donnie's 5.2. And
that's 5.2.
Where do I have
folks,
ours?
What we're looking at, just leave that up for a second, because what we're looking at there, folks, hang on one second. So what we're looking at there with the 5.2 is you're looking at 5 telomere repeats in one direction and then 2 in the opposite direction. So that's that head-to-head pattern, right?
You got a little thing in the middle, though.
Yeah, something's weird with that thing in the middle.
Yeah, and then this could have been one, but then it gets kind of weird around here. So, but then if you look at the this is the chromosome 2 fusion right over here, and you can see that there's a lot more repeats. It's 10 TTAGGGs and 41 CCCs. So we've got significantly more, basically.
I do want to highlight this comment. I honestly don't see why chromosome 9 is relevant here, even if it does turn out to be an ancient fusion.
I know.
It's not. It's
like,
not.
yeah, You found an ancient fusion site. You just proved telomere to telomere
Right.
fusion again.
Congratulations. Chromosomes
Yeah, thank
confused. We
you.
knew that. Yes.
I was excited when Donnie found that because I thought, yay, that that kind of tells us that we have to look for more of these things, right? That's pretty cool. Thanks, Donnie. Made our case for us. Anyway, what do I got here? Well, I got a picture of the actual DDX11 gene right here, or protein. And this is the DDX11
Yeah,
predicted
it's not the same.
L2. It's just, mostly disordered region here. And if you go ahead and look at, what else do I have? I got a lot. Okay, these are the domains, basically. DDX11 is out here on the end. It's got like one little tiny part of the ATP motor that drives this whole gene. And but these are all the domains. So if you look at where the domains are in this, I'm not sure what else I have here. I had a whole bunch.
Yeah, it's color code, folks. If you look at that figure, it's color coded to match.
Yeah.
Right. So you look at the sequence there, the colors match the protein, I believe.
Yeah. And that is from, let's see, I forget. I should have put the paper on there somewhere, but I didn't. But let's see. Let's go here. Let's see what else I got here. Let's see. Okay, that's the beginning. There's the gene. This shows you some of the domain. Oh, this shows you similarity and other things. These are, let's see. Okay, there's that figure again. And I've got a lot of stuff here. You have to reverse and do the reverse complement and all. But I mean, take a look at what I've done here. If you can't make
zoom in on that big one on the bottom, the big bars across the middle of your screen now that are all multicolored to the left of the black box. Yeah, that. Zoom in on that because I think that's kind of a good. What are we looking at there? Is that the same region?
I can't remember. Protein summary.
There you go.
I cannot remember. I think that's the, oh my God, I don't know what that is. And it's too fuzzy to even see. I forget what I was doing there.
So let's hit the high points for everybody. Make sure everyone leaves here with, getting the big picture across, everyone on the same page. So, Mike, what's the headline here?
Okay, the headline,
digging into the latest genome builds at the Chromosome 2 fusion site.
Every time you talk to these people, you want to talk about synteny, because it is perfect all the way across. And I'm going to dig even deeper into it. The second thing is it is very unlikely that exon was connected to a DDX gene. It's just a random little piece that got annotated, and we have since corrected it. And going forward into the future, we never need to talk about that exon again. There is no gene across the fusion site. There's no mystery. That is the bottom line, basically. All the current information tells us to leave that one by the roadside and forget it. But when they argue with you about chromosome 2, there's the biggest thing is to tell them about synteny all the way down. And that is the bottom line. It is undeniable. Now, either nature did it or God did it, but somebody slammed those two chromosomes together. We know that.
An analogy for this that I really like, that I remember reading probably between 15 and 20 years ago at this point, was from P.Z. Myers, where he said, if you have one car sitting at a stop sign and another car rear ends it, right?
Yeah.
And you've got the front bumper of the one car and the rear bumper of the other car, all messed up, right? And they're all smushed together and cracked up, and the radiators leak in and the trunk is whatever, right? Everything's, all the headlights and the taillights are broken. It's going to be hard to tell exactly what looking at that, what kind of car that is, right?
Yep.
But then you can look at the whole rest of the cars on either side of that impact zone, and you're going to get a lot of information about exactly what those two cars are. And that's the same thing here. When you have a telomere fusion, that necessitates having some breakdown of the telomere, because one of the functions of the telomeres is to prevent that thing from happening. But we can, in addition to finding the hallmarks of the telomeres there in a degenerate form, it's the stuff on either side, not just the immediate vicinity. but the entire length of that chromosome matching up with the two smaller chimp chromosomes.
Yep.
And that's synteny, and that's the whole point. And that figure right there is perfect. Yeah.
Yeah, and this you can see I'm documenting this in some detail. Here's the exact nucleotide where the fusion state starts. This is where it switches over from TT to CCC, and this is where it ends. And I'm going through and laying out all the genes. I got to work on the centromere here, this Anchor 3D. And but there's a whole lot of work that's been done here. And this is, let's see. Oh, we've got, this one lays it out in just mind-numbing detail about where the exons are.
The best kind of detail.
Fusion site, right? Yeah, I love it. I mean, I enjoy doing it. You really burn out doing this really fast, because I described it to my sister as I've got 7 anthills and I've got 3.5 billion ants in each anthill, and I want to find this this basically a group of 100 ants, and I want to find out how they compare to the other anthills. That's what I'm doing. And it's like, 3 billion ants is a lot of ants, but anyway. But there's more to come, and I'm hoping to do this. I'm hoping to break this into chunks and try to make it digestible, and show some of this stuff that I've been researching on my channel, and you're welcome to it,
which is linked below, and everybody should subscribe.
Answers in Atheism Before I Die, because I'm really old. Anyway, yep, that's a pile for the day. So
I dig it. That was great. I learned a ton from this. This is awesome. I didn't know any of those details about the ongoing revisions to the human genome, the reannotation, the removal of the exon from the far side of the fusion site. Fantastic. This is super cool. And then putting it together with the other genes in the vicinity. This is all great. I love this. Please keep doing it. And I've told you this privately, but I'm going to say it publicly. You should write this up, and I think everybody should pester you about that.
Yeah.
You should actually write this up.
I think I'm going to try.
You should try. Because that would be awesome.
In small pieces. And I don't know if anybody's still interested in chromosome 2, because we have just destroyed them. Oh God, I hate to use that word too. I hate it. Destroyed evolution, debunked and all that. I just, it's all these like high school fight words, it's just.
Let's put it this way. Creation should not bring up chromosome 2. It is a pristine example of a confirmed prediction.
Right.
And there's not just, again, it's not just one thing that confirms it. There's a whole bunch of independent things, each one of which is what we would expect in the case of a fusion. And we have talked about that here tonight.
The interesting thing that everybody has to know about what happens when you study evolution and you come to believe in evolution because you've studied it is every time you find a mystery or something that's out of place, you immediately start to say, oh my God, how did that happen? Right? You don't sit there and say, oh, I know evolution did it. I'm just going to discard it. No, you go to UCSC or NCBI or Ensemble and you dig in and you start trying to figure out how it happened. And I've been doing this for quite a while, and I will keep doing it every time. But it's almost like I am the biggest doubter of evolution that I know. Because every time I run into a mystery, I'll say, oh my God, how did that happen? Right?
Yeah.
And then you dig into it and you try to.
And then you spend months digging into it and you end up with all this information that we hadn't put together before. And this is great.
Yeah. Yeah. Then there's the other stuff about gene regulatory networks that we got to get into someday and talk about. And I know, what's his name? Zach
Hancock. Yeah.
Is really into that. And he glimpses what I glimpse. And what I'm seeing with these gene regulatory networks is this is how evolution happens, with this locking into these networks and bumping into these networks. And if you were to draw one of them, if you were to draw it in what it's actually doing, it would look like a big spaghetti bundle with a lot of dead ends. And every now and then, one of those dead ends matches up with another one, and kink, we got something that's happening, right? It either kills the organism, gets selected out, or it gives them a chance to reproduce a little bit better.
Now, Mike, people in the chat want your email address to be able to contact you. How can people contact you? You can put it in the chat. You can
put it in the chat.
I can put it in the description. Whatever you want to do.
It has my whole name at gmail.com, and try to use some title that I'm not going to think is junk. But
so, folks, I will get that email address and I will put it in the description of the video. Once we're done, I'll put it in the description. So if anyone wants to reach out to Mike, you will be able to do that.
Okay, that would be cool. Anyway, I hope I didn't embarrass myself.
That absolutely not. That was fantastic. I love hanging out and just like, let's crawl through some genomics. Let's do it. Yeah. It was great. So
yeah, I'd love to actually pull up the browser sometime and show you a little bit of that nightmare.
Yeah.
Because when you first get into that, you're lost. I mean, it takes a while to unwind it, but we could do a little bit of that too. But
all right, folks, the email address is in the chat right now. And I'm going to put that in the description once we are done. So if you need to reach Mike, that is how you can do it. Also, if you need to reach Mike, you should subscribe to his channel, which is linked down below. You should definitely do that. It's right there. Just do it right now. So, folks, this has been Mike. But how do I pronounce it? Bruzenak? Is that fair?
Yeah, Bruzenak. You don't have to pronounce you don't have to be real big on the E. Just Bruzenak.
Bruzenak.
Yep.
Mike Bruzenak, folks, doing research into human chromosome 2 fusion, which he should keep doing and write up, and it'll be great. And I want to see more of it.
There's so much more.
And you will see more of it on his channel, which again is linked down below, and you should subscribe. And you should also, if for some reason you stumbled in here, have no idea what's going on, you should also subscribe to my channel.
Subscribe anyway, and then you'll find out.
Subscribe right here, and you'll find out what is going on. What are we doing?
Right. Oh, folks, your channel is just so wonderful. I always you do such a good job of.
Thank you. You don't flatter. You don't have to do that. Don't do that.
Nah,
but I appreciate it. Thank you.
You need a pat on the back once in a while.
So yeah, for all the time creationists are yelling at me. I learned today apparently that if you Google my channel, the first thing that comes up is Discovery Institute complaining about me, which that makes me very happy.
They love to complain about you and Erica, and
that makes me very happy.
Yeah, it's wonderful. I only hope that someday they complain about me.
That is something we should all do.
Yeah, when I grow up, that's what I aspire to. When I finally grow up. So,
anyway. All right, folks, thanks for hanging out with us. This has been our meandering, our deep dive into the Chromosome 2 fusion site. And associated topics, and go to Mike's channel, subscribe to that, subscribe here, like it, leave a comment. I believe this chat is going to appear on Mike's channel at some point in the future.
Yeah, I'm going to premiere it. I'll download it and throw it up.
So for people in chat were like, this is really cool, but I don't know what this means, so I'm going to have to watch this again. Well, there you go. Go watch it again on Mike's channel.
Yeah, and feel free to ask me questions, and either pop into my chat or email me and ask me questions. And if 100 of you do it, I'm going to be in trouble. But if a few of you do it, I can handle it. So
go for it.
I've got
all right
600 unread emails today. So anyway.
All right. Thanks for being here, everybody. Hope you all have a great night.
Thank you.
Thanks again for sharing this with us. And keep an eye on Mike's stuff for more on this and other related topics. Catch everybody later.